I would first like to introduce Dr. Rosalyn Adam. She is the John Duckett Memorial Lecturer this morning. She is lecturing us on obstructive bladder disease, molecular insights, and therapeutic opportunities. She is currently the Director of Basic Science, Basic Urology Research, and Associate Professor at Boston Children's Hospital and Harvard Medical School. Please give her a warm welcome.
So good morning, everyone. I'd like to thank the organizers for the opportunity to present this John Duckett Memorial Lecture. It really is an honor and a privilege. I'd also like to extend my thanks to Paul Austin for the kind invitation to give this lecture. So in the next 15 minutes or so, I'd like to share some perspectives on the topic of obstructive bladder disease and to share some of our molecular insights and opportunities for new treatments. I have no disclosures. So my group has long been interested in the broad topic of pathological remodeling following obstruction, and notably the functional obstruction following spinal cord injury or disease, the ensuing bladder deterioration, and the search for treatments to prevent this. Although catheterization to promote bladder emptying and medication to reduce neurogenic detrusor overactivity and promote compliance provide some relief, these treatments are not universally effective. Moreover, there are no treatments thus far that specifically target the underlying fibroproliferative remodeling. Biomedical research in the 21st century has been revolutionized by our ability to perform high-content molecular profiling, as exemplified by initiatives such as the TCGA. This has provided mechanistic insights, opportunities for precision treatment. It has spurred the search for novel therapeutics, and it has enabled clinical advancements. However, the application of these approaches to benign urology, and in particular to obstructive uropathy, have proven more challenging. The bladder is not biopsied as part of routine care, so we lack tissue for analysis. Phenotypes may be mixed, and we lack high-quality reference data, especially for the bladder. So to address this gap, my group has used rodent models to explore the molecular landscape that occurs with obstruction and to evaluate new treatments. We have used a mid-thoracic spinal cord transection model that recapitulates salient features of neurogenic bladder pathophysiology, including neurogenic detrusor overactivity, profound remodeling with hypertrophy and hyperplasia, and matrix deposition consistent with that observed in human neurogenic bladder. So using this model, we expanded upon studies by a collaborator at Boston Children's, Larry Benowitz, who had demonstrated benefit of treatment with the purine nucleoside inosine on motor function in rats with spinal cord injury. Inosine is generated endogenously in our bodies by the deamination of adenosine, and based on studies in other organ systems, it's been known to have antioxidant, anti-inflammatory, neuroprotective, and neurotrophic activities. But at that time, its activity on the neurogenic bladder had not been determined. We proceeded to determine the potential benefit of inosine in neurogenic bladder, and in our rat model, we showed a significant reduction in detrusor overactivity in spinal cord injured rats receiving inosine versus those receiving vehicle. Notably, in the graph on the right, we found that inosine was effective at reducing NDO even after a 2-month delay in treatment. And so this suggested that it was effective in the face of existing damage. We proceeded to use our model to define the molecular basis and the molecular landscape that arises in the context of neurogenic bladder. So very briefly, we performed the spinal cord transection, as noted here. We maintained the animals for 8 weeks after injury and treated them either with inosine or vehicle daily for that time. At the conclusion of the experiment, we harvested tissue and urine, and we subjected them to either RNA sequencing for gene expression profiling or proteomics analysis using mass spectrometry. And then we proceeded to analyze and interpret those data. So to dive deeply into the molecular basis of the neurogenic bladder, we used an approach called causal network analysis. And simply put, causal network analysis allows us to answer the following question. So for a given gene expression profile, can we identify upstream regulators and effectors that would explain the gene expression pattern? And can we infer downstream biological impact? So when we applied this analysis to differentially expressed genes in the bladder tissue from injured animals versus controls, we identified inosine as the most significantly downregulated endogenous chemical after injury. Interrogation of our proteomic data identified an increase in an enzyme purine nucleoside phosphorylase, which converts inosine to hypoxanthine, thereby providing an explanation for the decrease predicted in our causal network analysis. Importantly, increased hypoxanthine and xanthine have been shown in other studies of lower urinary tract dysfunction to be associated with the induction of oxidative stress and other deleterious consequences. Additional findings from our model were an increase in oxidative stress, an increase in PARP activity, as evidenced by poly-ADP ribose signals shown in red here, and an increase in the DNA damage response, as evidenced by increases in gamma H2AX. Importantly, all of these were attenuated in animals treated with inosine. So to summarize, spinal cord injury is associated with a decrease in endogenous inosine, an increase in oxidative stress, an increase in DNA damage, and an increase in PARP activity, all of which were prevented by administration of exogenous inosine. These observations fit nicely with the paradigm put forth by Laurie Berder and Edmund Jackson, who have described the concept of uroprotective purines, such as adenosine, inosine, and guanosine, and urodamaging purines, such as hypoxanthine and xanthine, that are associated with the generation of reactive oxygen species and oxidative stress. Their observations have paved the way for studies of PNPA's inhibition using a compound called 8-aminoguanine to shift the balance towards a more protective purine milieu. Collectively, our studies suggest that maintenance of endogenous inosine levels, whether by inhibition of breakdown or by administering inosine exogenously, is beneficial for bladder health. So how does this relate to neurogenic bladder and functional obstruction in human patients? So to determine first whether the changes in tissue are reflected in urine, we collaborated with Richard Lee and John Freilich in our institution to assess the urinary proteome in both our rodent model and pediatric patients with neurogenic bladder. And when we compare the proteomes, we find that the urine profile of pediatric patients clusters with that of the rodent, providing confidence that the rat model is informative with respect to obstructive bladder disease in humans. We also proceeded to assess urine from our rat model in animals treated without and with inosine. And what we find here are a number of pathways that are predicted from the differentially expressed proteins. These are predicted to be enriched with injury, all of which were reversed in the urines from the rats treated with inosine. I won't go into the details here, but a number of the pathways that are enriched were also identified in our omics analysis of tissue, suggesting that alterations in tissue are reflected in the urine, and they're also sensitive to treatment, providing a valuable source of biomarkers for both disease and response to treatment. So what have we learned from this analysis? Well, we've identified new insights into the molecular landscape of the bladder following functional obstruction. We've identified novel, previously unappreciated targets for intervention. We've provided evidence for inosine as a potential treatment. And we've also highlighted the utility of urine biomarkers to both inform disease progression and the response to intervention. So with that, I'd like to conclude by acknowledging the contributions of many colleagues at Boston Children's Hospital, our collaborators in Boston and beyond, our funding sources, notably from the PBA Research Foundation, the National Institutes of Health, the Department of Defense, and the Department of Urology at Boston Children's through the Children's Urological Foundation. Again, I'd like to thank the organizers for the privilege of giving this lecture, and thank you for your attention.